All right, welcome everybody. You've heard a lot about me, and you're going to hear a bit more. And today I'm going to talk broadly about my research career and trajectory and how I apply the tools of genetic epidemiology to investigate neuropsychiatric conditions. So a bit about the Alma C Lab. You just heard Dr. Alma C. So our main aim is in the development and application of statistical genetic methods for the localization, identification, and characterization of genetic effects on common complex disorders and related quantitative risk factors. So she has worked both in pediatric and adult studies, family and population cohorts, and looking at normal variation in brain structure and function. And in addition to the neuropsychiatric conditions, she also works in cardiovascular disorders, but I do not have that specialization. So the main mission is, how do we understand human variation? How do genes in the environment interact to make us who we are? Why are some people different from others? And are the same factors that influence variation in the middle of the distribution the ones that push people out to the tails past the threshold of disease risk? Now, specifically, the themes of my research, as she said, is look at the multifactorial risk factors for psychiatric disorders as they emerge across development. This work is the culmination of my training and experience in clinical assessment, epidemiology, and genetics. But why mental health? And I'm sure we've all seen slides like this before. I'll define.
If you want to wear that.
Do you want me to wear that?
Yeah.
Okay. I'm going to wear a microphone. This way. Okay. So this shows the global burden of non-communicable diseases in disability-adjusted life years worldwide. So the DALY is a measure of the overall disease burden expressed as the number of years lost due to ill health, disability, or early death. And you can see that neuropsychiatric disorders are the largest proportion of all the non-communicable diseases, and that encompasses schizophrenia, affective disorders, bipolar dementia, substance, and other mental disorders. But the major contribution of psychiatric epidemiology over the past decade has been its demonstration of the dramatic impact that these disorders have on the individual who suffers the mental disorder, his or her family, and the huge economic impact on society. And within the mental disorders specifically, depressive disorders are the largest contributor. Depression is one of the leading causes of disability worldwide and has the highest impact of all mental health and substance use disorders. Bipolar disorder also has a tremendous impact on lifelong disability that is equivalent to that of schizophrenia, alcohol use, and drug disorders combined. The burden of mental and substance use disorders is growing with a 37.6% increase between 1990 and 2010, and that's just continuing. Now, just the economic cost of these things. So this is again another way of looking at the DALYs. You can see that mental and behavioral disorders are second only to cancer and cardiovascular disease as the third leading cause of disability in the U.S. overall. And it's projected that depression costs us $233 billion a year. And that includes the lost productivity and treatment and other costs associated with it. I don't know why it's in 2016 dollars. Oh, because it was in 23? Anyway. But why do we look at youth? Mental health disorders across the lifespan are shown here as a cumulative incidence plot. And you can see that 70% of mental disorders onset prior to the age of 25. So there's been a changing pattern of childhood morbidity. There's an increase in chronic health problems over the past few years. And a greater prevalence of mental health disorders from 15 to 22% of the population. There's a greater appreciation of the role and impact of neurodevelopmental health problems, specifically learning and language, growing from 10 to 17% of the population. And then there's a growing number of children with multiple conditions, including asthma, obesity, ADHD, and so forth, that I'll talk more specifically about with my research later in the presentation. So why are these disorders so prevalent and so complicated? Well, we don't know what causes them. The unknown etiology is what we're trying to elucidate in all of our research. We have a lack of valid biologic trait markers for these disorders. There's heterogeneity in both the causes and the manifestations of these disorders. They're dimensional, and we tend to cap them in categories. There's a lack of validity in the classification. That's imprecise boundaries and thresholds. So whether you see a doctor at one point, they'll tell you have bipolar, and another time they'll tell you have schizophrenia or anxiety and depression. But that is also complicated by maybe those conditions just happen to coexist more often than not. And that's why I talk about comorbidity within classes of mental disorders and between mental disorders and physical conditions. But the situation is not unsurprising in light of the multifactorial nature and complexity of them. You know, it's not as simple as taking a blood test and seeing what's going on. And also, it's not expressed here, but measuring the environmental impact that leads to the development of these conditions is incredibly challenging. So my background and training. Dr. Alma C gave you a brief view of that, but I wanted to start by saying I didn't set out to do any of this. I ended up at Carnegie Mellon because I wanted to be an opera singer. And now I've ended up here in an informatics speaker series, so I kind of like to tell that to all the kiddos that, you know, it's not set out. You go the way you go and you end up where you end up. But I went there to sing and ended up taking a class in cognitive psychology my freshman year of college. And it was so exciting that it changed everything that I was doing. And I'm going to point out this guy up on the top right is Joel Greenhouse, a professor of statistics at Carnegie Mellon. I never would have taken even a statistics course had it not been required in my core curriculum for psychology. And all of a sudden I'm doing computer science and statistics and loving it. Again, my 8th grade algebra teacher told my mother that sometimes girls just can't do math. So, you know, things change. Anyway, I got my degree in psychology, cognitive psychology at Carnegie Mellon, and I didn't know what to do with myself after that, graduate school, medical school, and I went to the post-baccalaureate fellowship training program at the National Institute of Mental Health and worked with this guy over here, Danny Pine, who runs the section for SDAN, Development and Affective Neuroscience. We did neuroimaging studies on kids with anxiety, depression, and typical development. And working with him, I attended graduate classes at night and pre-medical coursework at the Foundation for the Advanced Education in the Sciences, which is the graduate school at the NIH. And I took an epidemiology course with a doctor who had worked for 25 years on the Framingham heart study. And that's where I fell in love with epidemiology. And so I went down the road to George Washington, got my master's in public health and epidemiology, and worked at the CDC, National Center for Health Statistics, for my practical internship. And that's where I was like, we look at all these risk factors, and the epidemiologists look at the environmental risk factors, but they don't account for the genetic variation, which is the largest chunk of non-modifiable risk. So how do we quantify that? That led me to do a PhD in neuropsychiatric genetics in Trinity College in Dublin. That's a longer story. We don't need to get into all of that today. So what is epidemiology? Just to make sure that we're all on the same page. It's the study of the distribution and determinants of disease or health-related states. It's not just ill health. It's also, you can use it to investigate wellness. What things make people happy and healthy and have good lives. And in specific populations, a lot of people think epidemiology has to be a whole representative sample of the population at large, but you can look at the neighborhood level, a specific school, city, state, country, or globally. And the major goal of epidemiology is to identify the etiology of disease and thereby prevent or intervene in the progression of the disorder. We like to say it's more than just counting. And so when I was working at the National Center for Health Statistics, with Dr. Cleary here on the right, who is a global health epidemiologist who actually did all sorts of cool stuff in Nepal, and it was a Fulbright he did in Nepal. And Pauline Mendola, who's on the left, who was at the National Center for Health Statistics at the time, and she is now the director of epidemiology at the National Institute for Child Health and Human Development. I did a study in the NHANES, which is the National Health and Nutrition Examination Survey, which is a population-based study of the United States, where they do a full psychiatric interview and measured height and weight. And I looked at the association between major depressive disorder and obesity in US adolescents. This was a complicated study design because we had to use sampling weights from the population to take, because they oversampled underrepresented minorities so that we could get good rates of disorders in those unrepresented groups. And we had assumed that the association between depressive disorder and obesity would be most strong in adolescent girls because of the societal impact of weight and not social media at this time, but Maxim magazine or those sorts of things. But that's not what we found. So just to orient you to this, here we're looking by sex, age, race, ethnicity, and poverty income ratio, because there is a strong association with poverty and obesity. Just ill health generally is associated with poverty. So when we did our adjusted logistic regression models, we included age, sex, race, ethnicity, and poverty in our models. And what we actually found, that it was males who had an increased risk of obesity with depression, and it was non-Hispanic blacks. So the rate of depression in the sample was actually quite low. It was 3.2% in this cohort of youth, and only 1% among males and nearly 5% among females, which is more in line with what we would think post-pubertally. But that's why I point that out, because depression changes over the lifespan by sex. It is much more common in women than men, but that is only after the point of puberty. Obesity was much more common, and then actually this terminology is interesting as well. I think now we might have to say at risk for overweight, because obesity was stigmatizing language. I learned a lot about bureaucracy getting this paper through clearance at CDC. But we don't have a good explanation for why we had this finding, and I haven't been able to follow it up yet. But it's an easy thing for health professionals to say, like, overweight, maybe we should do more health screening or mental health screening than we otherwise would. But then we move on to the genetic epidemiologic research. So what is the point of that? We want to establish that there is a genetic component to a trait or disorder before we undertake genetics research to look at that. So genetic epidemiology is the epidemiological evaluation of the role of inherited causes of disease in families and in populations. So how we do that is we establish first that there is a genetic component, then establish the relative size of that genetic effect compared to other sources of variation in disease risk. And those can be environmental effects, physical or chemical effects, or even behavioral or social aspects of disorders. And then from there, we move to identifying the genetic variants responsible for the genetic component. And I like this widely accepted definition by Morton, is that genetic epidemiology is a science which deals with the etiology, distribution, and control of disease in groups of relatives with inherited causes of disease in populations. It's very wordy, but I think it gets the point across. So how do we study this? What is the contribution of genetics and environmental factors to a disease or trait? So first, family studies compare the aggregation of a disease in relatives of cases with a disease, cases with a disease compared to controls without the disease. We can look at twins and adoption studies, which give us information about the relative contribution of genetic and environmental risk factors. And migration studies have provided information about environmental exposures in genetically similar cohorts. So you look to see the rate of disease in women in China and migrant Chinese women to the United States, for example. And so family studies, we've all seen these pedigrees, and the question is, are you more likely to see a disorder in a family member of someone with that disorder or not? And the recurrence risk is the chance that the disease will strike again. In medical genetics, the chance that an inherited disease is present in a family will recur or affect other persons. Recurrence risk is one of the things you'll see in the literature. And I like this little diagram of twin studies. So we have monozygotic twins on the left and dizygotic twins on the right. And the assumption is that twins are exposed to the same environmental risk factors. The second assumption is that identical twins, the monozygotic twins, share the same genome, which is probably not entirely true, but close enough for now. And that fraternal twins or dizygotic twins share on average 50% of their genomes. So you can compare you can calculate the concordance in diagnosis. So you measure the concordance in the dizygotic pairs and the concordance in the monozygotic pairs. And if the concordance in the monozygotic pairs is greater than the concordance in the dizygotic pairs, it is indicative of an increased shared genetic risk. So then, what are some other methods we use to identify genetic variants? We can complete segregation analyses, and that looks like what does the genetic component look like? What is the model of transmission? Is it dominant? Is it recessive? Is it sex-linked? And to do that, you need multi-generational family studies, preferably with more than one affected member in order to define the pattern. You can do linkage studies. Where is the location of the disease gene? And for that, we use segregation of the genetic locus with the disease phenotype. Or we can do association studies. So what is the allele associated with disease susceptibility? And you can do that in case control or within families. But DNA variation comes in many forms. You can have changes of a single base pair. So that's shown here. So SNP is single nucleotide polymorphism. And you can see that here, this person has an A, this person has a G, and this person has a T. So, and if that variation occurs more than 1% frequency in the population, we call it a single nucleotide polymorphism. Now, you can also have single base pair insertions or deletions. So you see here, this T has been inserted where it's not up here, and here this A has vanished from our code here. It results in extra genetic material or loss of genetic material, and then as that DNA is transcribed and translated and transcribed, you can get altered protein products. It has been generally assumed that deletions have more of an impact than duplications because it's easier to downregulate too much of something than it is to make up for the absence of something. And then we have deletions or duplications of entire segments of DNA. So here you see that this section is missing from this chromosome, or here this section is copied twice. And the same theory applies for the deletion being more severe than the duplication. These sort of mutations can be inherited from your parents, or they can be de novo. That means they develop in the individual. And at least when I was doing my PhD work, it was assumed that these, which are collectively called copy number variations, occur in 12 to 15% of the human genome. And some of those are recurrent, you see them in lots of people, and some of them are individual. And they can have strong phenotypic effects, such as the 22q1 deletion, or they can be unidentified for your entire life. So when I went to Dublin, this was at the outset of copy number variation research in neuropsychiatric disorders. Jonathan Sabat had just published a paper looking at copy number variation in autism spectrum disorders. It was the first publication to look at that. And I put in a grant to the Irish government to look at copy number variation broadly in neuropsychiatric disorders. And I was lucky to receive that. And so the first thing I set about doing was a review of what had been found to date in copy number variation research in neuropsychiatric and neurodevelopmental disorders. We focused specifically on autism and schizophrenia, where the majority of research had been completed. What's interesting is this built on my interest in mental health, but expanded to disorders that I didn't have experience with. I'd worked primarily in mood and anxiety disorders, and here I was transitioning to developmental disorders. And this review was published in Trends in Genetics. And the main findings that we had were, if you look at this karyogram, so each of these, it's the chromosomes from 1 down to 22. We don't have the sex chromosomes here because they're difficult to study and people leave them off, which I think is a problem given the sex differences in neuropsychiatric conditions. But for reasons I won't go into, people are working on this. But what was interesting is that you can see the regions of the genome in red, which are regions that had been implicated in more than one study in autism. The regions in blue are regions that had been implicated in schizophrenia, and these regions in black had been implicated in both disorders. Now, its example is the 16p11.2 chromosome and the 22q deletion region have been found in multiple studies, and people were aware of this. But the question is why? So we set out to look at the phenotypic manifestations of these rare CNVs in autism spectrum disorder. I did a similar study in schizophrenia, but I'm going to concentrate on the autism disorder here. Through this project, I was the coordinator for the phenotype sort of harmonization of the Autism Genome Project, which was 50 research groups in 19 countries. So just to put it into perspective, there were, I think, 47 different IQ tests, and there was a committee that I was not on that harmonized this so that we could get singular measures of verbal performance and full-scale IQ in these kids. And I'm thankful to the team that did that. But I was just trying to make sure we got all the data from all the sites and then coordinated it. And then because we were looking at rare events, we needed a way to aggregate across CNVs. You can't do statistical tests on 3 things. So we created 2 ways of looking at the aggregating the CNVs so we could do statistical analysis. And I'll flash up the findings just so I can show you. So on the top left, you see autism and intellectual disability CNVs. What those were CNVs that had been previously implicated in autism or in intellectual disability. It was about 101 CNVs that were curated in the literature. And we aggregated across those and compared people who had those CNVs with people who didn't have those CNVs but had other CNVs. We also looked at whether or not they were de novo or inherited. And then as a comparator group, we looked at all CNVs in genes that were brain expressed. And that had been defined by a group at the Broad where they'd taken data from large expression atlases. This was before GTEx was online. So they'd curated the literature. So I'm sure there are all sorts of biases and problems with that list, but I'd be interested to see how this analysis would come out now. But it was about 3,000 genes that were shown to be differentially expressed in brain and central nervous system tissue compared to other tissues. And we looked again at whether those were de novo or inherited. And then the bottom line I'll come to in a minute. But what we found overall was that autism and intellectual disability CNVs were associated with language and communication phenotypes, which was pretty much what we expected. But we found that when these CNVs were de novo, they were associated with whether or not the child was verbal and whether or not they had seizures, which are more parts of the syndromic form of autism or more severe form of autism. So rather than the inherited variety, maybe these de novo CNVs were more severe. With the brain expressed duplications, we found that that was actually associated with better adaptive function on the Vineland scales. And we don't really know why that is, but it was a strong effect. We looked at de novo brain expressed CNVs, and they were associated with advanced paternal age and simplex family type, meaning that only one child in the family had autism. And that's why I'm going to get to advanced parental age. Here, that was associated with brain expressed CNVs overall. There's a theory that advancing maternal and paternal age leads to more new mutations, which leads to more severe disorder. That was the theory. It has actually not been tested directly, this chain of events. But we did find that increased parental age was associated with these CNVs. We also did other machine learning and modeling techniques. So one of the other things I did was a random forest analysis, which is a type of regression and classification algorithm. And it's called a forest because you do regression trees. You give it a splitting variable, and then it resamples the data and tells you the best predictor for that phenotype. So here I split the data on whether or not someone had the autism intellectual disability CNV or not, and gave it all the phenotypes. And here the best predictor for whether or not someone had one of these CNVs was the verbal IQ. And so that linked back well with our findings from the standard frequentist analysis that the ASD-ID CNVs, I could acronym all day, were associated with language disorders. But you can see the ones that are negative were actually variables that hindered classification. Why this is called a forest is you run a tree, but you run 10,000 trees, and you leave an out-of-bag sample. So it might be difficult to see on the bottom right here, the OOB error rate. That indicates that the holdout set, which here we used was 1/3 of the data, was misclassified using the previous classifier 6% of the time. So it actually did pretty well. And here, the majority of the variables we selected assisted in classification, but some of them did hinder it. And so if you were following up things, you would leave out those ones that were hindering classification. I also did some latent variable analysis and other interesting modeling that I would be happy to speak about some other time, but it could take the whole talk just to explain those bits. So when I finished my PhD and I decided what to do for a postdoc, I actually came here to Penn, as Dr. Alma C said, and worked in the Gore Lab in Neuropsychiatry. And I wanted to follow up some of these parental age findings, but more globally. Instead of just looking at autism, I wanted to look at psychopathology broadly. Did parental age, maternal and paternal age, have an impact on the development of psychiatric conditions in youth? And to do that, I used data from the Philadelphia Neurodevelopmental Cohort, which many people in this room may be familiar with. But just to give it briefly, it was a sample that was recruited here between CHOP and Penn. It's nearly 10,000 youth ages 8 to 21, and they all underwent a comprehensive screening for mental disorders. There were chronic physical conditions reported by the parent or the youth, and there was an electronic medical record review. All of the kids were genotyped here at CHOP, and then the data was imputed on the Michigan imputation server, hard calls generated. I could go on if you want details about the genotyping. I'd be happy to talk about that later. But the study was designed to assess behavioral dimensions reflecting vulnerability to major mental illnesses. And but what I did with that here, this part of the study doesn't use the genetics, but I just wanted to say that for later. Here I looked across disorders, and I'll orient you to this plot. So these are maternal age on the left and paternal age on the right. And sorry, I'm at an awkward angle. So the increasing age, 20 to 60 on both sides. And then down here on the bottom, you can see it's no disorder and then the disorder. And by comparing these little notches, this is actually the 95% confidence interval in those notches. So if they don't overlap, you have a statistically significant association. So I observed associations between both younger and older parental age with different psychiatric conditions. So specifically in the fully adjusted models, these are here just the raw numbers that you can't plot the fully adjusted models. Maternal and paternal ages were associated with behavior disorders and psychosis, and that was both younger maternal and paternal age. So you can see that. In the psychosis, it's probably the most obvious here. So this is kids with no psychotic symptoms and kids with psychotic symptoms. But it's worth noting that the width of the bars here indicates the sample size. So we have very few kids who have psychosis symptoms compared to kids who have anxiety disorders. So it's just something to consider. So we found younger parental age for the psychosis, which was surprising, and for behavior disorders, which was not. And then we did find an association between advanced maternal and paternal age with, here it's the category is, it's listed as PDD, which in our cohort was pervasive developmental disorders or autism spectrum disorder. And it was a single question. Has a medical professional told you that you have this? So it's not a full assessment, but we did go on and do case validation through the medical records, and were able to validate more than 2/3 of those reported diagnoses. And so then after completing that and other work in the Gore Lab, I came to work with Dr. Alma C here at CHOP. And I'm continuing to analyze data from the PNC, which is quite convenient, examining risk factors for child and adolescent psychopathology. But the primary thing I'm doing now is investigating medical mental comorbidity and the association between genetic variants and empirically derived subgroups of psychiatric and somatic disorders. So why look at medical mental comorbidity? Comorbidity may be due to loci or biological pathways in the etiology of both conditions. Identifying these loci may provide insight into etiology. And so we were interested, is there a common genetic susceptibility to comorbid mental and physical disorders? Because 25% of people have both disorders. So I got a grant, you can see it up here, from ITMAT, the Institute for Translational Medicine and Therapeutics here. That's a bridge between CHOP and Penn. So I'm completing this work in the PNC, and my colleague Rachel Kember is completing work in the Penn Medicine Biobank, which is an adult sample from Philadelphia, and we're going to compare developmentally across the kids and the adults, the medical mental comorbidity and the genetic architecture, and see if it's the same. So as I said briefly, is it a common risk factor or common risk factors collectively that contribute to medical conditions and psychiatric disorders? are there behavioral and environmental factors, such as psychosocial stress, that lead you to have cardiovascular disease and depression? Or is it a biological mechanism, like global measures of inflammation are associated with cardiovascular disease and depression? Or is it that one disorder actually causes the other disorder? And to do that, you have to have the temporal association, which is harder to do in our cross-sectional studies. And unfortunately, I can't do that here, but we can start to look at that in the biobank, which has longitudinal medical records. So what I did here was look at patterns of medical mental comorbidity across development with standard regression techniques. And then I looked at models of these polygenic risk scores, which is an aggregate measure of common genetic risk, to see if those polygenic risk scores for a psychiatric disorder predicted or were associated with, rather, a medical condition, or whether they were polygenic risk scores for medical conditions were associated with their comorbid psychiatric disorders. And just to show the depth of comorbidity in the PNC, this is for the entire sample, not the subset that's genotyped. And the comorbidity is pervasive. I do want to point out that this anxiety disorder number is incredibly high, and that's because in this plot I've included simple phobias. If you remove simple phobias, the rate of anxiety disorders drops to standard population levels. And our behavior disorder rate is also very high, but we think that's a result of sampling. So for the comorbidity between psychopathology and medical conditions, we had system-level medical conditions because they've been aggregated. So these are not specific disorders that come down on the left-hand side here. But I wanted to point out this overall medical rating, which is a rating of overall medical severity, because in each of these categories, people can appear more than one. They don't appear more than once in the category, but they can appear in multiple categories. And here we found that if we look at overall psychopathology and overall medical conditions, there is an association. It's weak, but present. But we have strong associations for psychopathology and developmental conditions. Developmental conditions here include a whole host of things. So it's a very umbrella term. In bedwetting, language delay, reading delays, all sorts of learning problems, and some motor development issues. Whereas the CNS here encompasses a whole host of things: meningitis, multiple sclerosis, epilepsy, and headaches. And then we found, I didn't go into it here, but the developmental condition association with psychopathology was present in all of the psychiatric disorders we looked at. And strangely, ear, nose, and throat was associated with psychosis symptoms, but you see how wide this confidence interval is? So it's because the numbers are very small, so I'm not sure what to make of that. And then what we looked at was, and I know this can be quite confusing, here on this side are the diagnoses in the cohort, and this is the overall psychopathology measure. And on the left side are all the polygenic risk scores that I was able to test. And this is in proof of concept stage. So we just wanted to see if the polygenic risk score for anxiety predicted anxiety in this cohort. And it does not, for example. The ones that did work, so to speak, were that the ASD, or that's not ASD, the ADHD polygenic risk score did predict or was associated with ADHD and behavior disorders in the PNC. And the major depressive disorder polygenic risk score was associated with mood disorders broadly in this cohort. But the bipolar disorder polygenic risk score was not. So this is, I think, clearly a depression association. The schizophrenia polygenic risk score was not associated with psychosis symptoms, but that's also not surprising because these are children who have not yet developed schizophrenia. They are on the spectrum of psychosis symptoms. So it's not quite the same thing, and certainly not all of them will convert to schizophrenia. And I was interested to see that the cross-disorder polygenic risk score, which includes results from 5 different genome-wide association studies of mental disorders, was not associated with overall psychopathology in this cohort. So I have to drill down on what that is. But also as a proof of concept, because there had been previous association of mood disorders, we wanted to look at a number of autoimmune conditions. Because I had the polygenic risk score data available, we're trying to track down more to do this with the actual comorbid conditions we have. But we found that none of these polygenic risk scores for these medical conditions were associated with mood disorders in this cohort, which was sort of surprising. So in summary, this is the first study to examine the genetic architecture or common genetic architecture underlying comorbid medical and mental conditions in a large and diverse sample of children. Granted, the genetic analyses were only done on the European subset because of limitations of polygenic risk scores at this time, but that's changing as we speak, I think, although slowly. And the specific patterns of comorbidity between physical and mental conditions have important implications for etiology, but we don't yet know what those are. And the overlap is not well explained by this common genetic variation, using polygenic risk scores as a proxy for common genetic variation. So what we're going to do moving forward in this study is derive empirically based subgroups of conditions using some of the machine learning techniques I talked about before, and perhaps some new ones, and get the data to calculate polygenic risk scores for specific medical conditions in the classes that we had. Also, we're going to do prospective tracking of cross-disorder comorbidity. So in conclusion, understanding the etiology can inform treatment and prevention, but it's going to take us some time and teamwork to get there. And that's why I say teams of researchers with expertise of clinicians, biostatisticians, geneticists, and molecular biologists are required to complete this type of research. You need clinicians to get your patients and inform your clinical questions. You need biostatisticians to help you do the appropriate analyses, and geneticists to do the genetic data, molecular biologists to help you look at the fundamental underlying mechanisms, and we all need to work together. So larger collaborative studies and with perspective and comprehensive phenotyping will empower researchers to do that. You need to know what comes first and in order to figure out the chain of causality. And that will help us to combine the complicated research of genetics and environment. And so I have a lot of people to acknowledge. All of the individuals and families that participated in the research project. I have done studies now with participants in the more than 100,000, so I couldn't possibly thank all of them. All the members of the recruitment team, particularly at the Penn Brain and Behavior Laboratory, who recruited and assessed 10,000 kids in 2 years, which I think is pretty impressive. And then all the mentors who've helped me along the way. Like, I wouldn't be here without the support of all those people. There's specific people that worked on the projects that are listed here. And then, of course, all the funding, because we can't do this without money. And I'm apparently supposed to say that this content is solely my responsibility and does not reflect that of the NIH. And with that, I want to thank you for your attention.